The overall goal of this procedure is to determine the transcriptome-wide binding sites of RNA-binding proteins, or RBPs. This is accomplished by first using photoreactive nucleoside analogues for in vivo labeling of nascent transcripts. The second step of the procedure is to crosslink RNA and RNA-binding proteins by 365 nanometer UV light. The third step of the procedure is to immunoprecipitate the investigated RNA-binding protein and to recover the cross-linked RNA. The final step of the procedure is to convert the RNA into a cDNA library and deep sequence it. Ultimately, results can be obtained that show transcriptome-wide binding sites of the RBPs and enable the definition of the RNA recognition element and its binding preferences.
Hi, my name is Markus Hafner from the lab of Tom Tuschl at the Rockefeller University in New York. Today we'll be showing you a method called PAR-CliP, or Photoactivatable Ribonucleoside-Enhanced crosslinking and immunoprecipitation. We use this procedure in our lab to identify on a transcriptome-wide scale binding sites of RNA-binding proteins and their binding motifs and preferences. Okay, so let's get started.
In vivo UV crosslinking begins in a laminar flow hood. In this protocol, we will use tagged IGF-2 BP1 cells as an exemplary model. Using sterile technique, expand HEK293 cells stably expressing flag HA tagged IGF-2 BP1 under control of a TET repressor in HEK293 cell culture medium. Use between 100 and 400 times 10 to the 6th cells and grow them to approximately 80% confluency. At 14 hours before cross-linking the cells, add the photoreactive ribonucleoside analog 4-thiouridine, or 4-SU, directly to the cell culture medium. Alternatively, 6-thioguanosine, or 6-SG, can be used. Following addition of the photoreactive ribonucleoside, induce expression of the flag HA-tagged IGF-2 BP1 by the addition of doxycycline. Cells are then incubated overnight in cell incubator. Next, decant the growth medium. Then wash the cells once with 10 milliliters of ice-cold phosphate-buffered saline per plate. Then remove the PBS completely by decanting. Place the uncovered plates on a tray with ice and irradiate them with 365 nanometer UV light in a Stratolinker 2400 UV crosslinker, or equivalent. Subsequent to UV irradiation, scrape the cells off with a rubber policeman in 1 milliliter of PBS per plate. Transfer the cells to 50 milliliter centrifugation tubes and centrifuge at 500 G for 5 minutes at 4 degrees Celsius. Discard the supernatant and retain the cell pellet. At this point, cell pellets can be flash frozen in liquid nitrogen and stored at -80 for at least 12 months. To continue with the procedure, resuspend the pellet in 3 volumes of 1x NP40 lysis buffer and incubate on ice for 10 minutes. Clear the cell lysate by centrifugation at 13,000 G for 15 minutes at 4 degrees Celsius. Following centrifugation, clear the lysate further by filtering it through a 5-micron membrane syringe filter. Finally, add RNase T1 to a final concentration of 1 unit per microliter and incubate in a water bath at 22 degrees Celsius for 15 minutes. Subsequently, cool the reaction on ice for 5 minutes. Following in vivo UV crosslinking, prepare the magnetic beads for immunoprecipitation. Transfer 10 microliters of Dynabeads protein G magnetic particles per 1 ml of cell lysate to a 1.5 milliliter microcentrifuge tube. Then, place the tube containing the beads on a magnetic stand for 1 to 2 minutes. Remove the supernatant and add 1 milliliter of citrate phosphate buffer to the tube while it is on the magnetic separator. Next, cap the tube and vortex the sample to resuspend the beads. Centrifuge briefly to collect any beads remaining in the tube cap. Repeat this wash with an additional 1 milliliter of citrate phosphate buffer. Following the wash, resuspend the beads in citrate phosphate buffer at a volume of 2 times the original volume of bead suspension. Then, Add 0.25 milligrams of anti-flag M2 monoclonal antibody per 1 milliliter of bead suspension. Incubate the suspension on a rotating wheel for 40 minutes at room temperature. After the incubation, wash the beads twice in 1 milliliter of citrate phosphate buffer as previously described. This wash serves to remove any unbound antibody. Following the wash, resuspend the beads in citrate phosphate buffer as before. To begin immunoprecipitation, add the freshly prepared antibody-conjugated magnetic beads to the RNase T1 treated cell lysate. Transfer the mixture to 15 milliliter centrifugation tubes and rotate for 1 hour at 4 degrees Celsius. Following incubation, collect the magnetic beads, discard the supernatant, resuspend beads in 1 milliliter of IP wash buffer, and transfer them to 1.5 milliliter microcentrifuge tubes. Wash the beads as described before, 2 times in 1 milliliter of IP wash buffer. After the final wash, resuspend the beads in the original bead volume of IP wash buffer. Then, add RNase T1 to a final concentration of 100 units per microliter. Incubate the bead suspension in a 22 degrees Celsius water bath for 15 minutes. Subsequently, cool the suspension on ice for 5 minutes. Next, wash the beads 3 times in 1 milliliter of high salt wash buffer. Then, resuspend the beads in 1 original bead volume of dephosphorylation buffer. Add calf intestinal alkaline phosphatase to a final concentration of 0.5 units per microliter and incubate the suspension for 10 minutes at 37 degrees Celsius. Wash the beads twice in 1 milliliter of phosphatase wash buffer, and then twice more in polynucleotide kinase or PNK buffer without DTT. Finally, resuspend the beads in 1 original bead volume of PNK buffer. To begin radiolabeling of cross-linked RNA, add gamma-32P ATP and T4 PNK to 1 original bead volume of the freshly prepared bead suspension. Incubate the suspension for 30 minutes at 37 degrees Celsius. Next, add non-radioactive ATP to the suspension and incubate it for another 5 minutes at 37 degrees Celsius. Then, wash the magnetic beads 5 times with 800 microliters of PNK buffer without DTT. Finally, resuspend the beads in 70 microliters of SDS page loading buffer. Incubate the radiolabeled suspension for 5 minutes in a heat block at 95 degrees Celsius to denature and release the immunoprecipitated RNA binding proteins with the cross-linked RNA. Then, remove the magnetic beads on the separator and transfer the supernatant to a clean 1.5 milliliter microcentrifuge tube. Load 40 microliters of the supernatant per well of a Novex bis Tris 4 to 12% precast polyacrylamide gel and run the gel for 55 minutes at 200 volts. Once the gel has run, disassemble the gel chamber and dismantle the plates, leaving the gel mounted on one plate. To facilitate the alignment of the gel to the phosphoimager paper printout, implant 3 tiny radioactive gel pieces asymmetrically at the gel corners. Wrap the gel in a plastic film, such as Saran wrap, to avoid contamination. Expose the gel to a blanked phosphoimager screen for 1 hour and visualize it on a phosphoimager. Next, align the gel on top of the phosphoimager printout using the implanted gel pieces for orientation. Cut out the bands that correspond to the expected size of the RNA binding protein, or RBP. Transfer the bands to a D-tube dialyzer MIDI tube and add 800 microliters of 1X SDS running buffer. Electroelute the cross-linked RNA-RBP complex at 100 volts for 2 hours, per the manufacturer's instructions. Subsequent to the electroelution, add proteinase K buffer and proteinase K. Incubate the solution for 30 minutes at 55 degrees Celsius. Recover the RNA by acidic phenol chloroform isoamyl alcohol extraction, followed by a chloroform extraction. Then, add 1 microliter of glycogen and precipitate the RNA by adding 3 volumes of ethanol. Finally, dissolve the resulting pellet in 10.5 microliters of water. Once the RNA has been recovered, carry it through a standard cDNA library preparation protocol and perform careful bioinformatic analysis of the sequence reads. Representative results for a PAR-CliP performed with HEK293 cells expressing flag HA-tagged IGF2BP1 are shown with the photoreactive ribonucleoside analogues 4SU and 6SG. Crosslinking efficiency of the alternative photoreactive ribonucleoside 6SG is generally lower than for 4SU. The lower crosslinking efficiency might result in a higher background of sequences derived from fragments of abundant cellular RNAs, and therefore more cells should be initially used for the experiment. Different PAR-CliP photoreactive uridine analogues are compared to traditional UV254 nanometer crosslinking, or CLIP. As seen here, UV254 nanometer RNA protein crosslinking has limited crosslinking efficiency. The intensity of the appropriately sized radioactive band indicates whether the PAR-CliP experiment has worked and whether sufficient RNA has been isolated to carry through a small RNA sequencing protocol. The frequencies of characteristic mutations in the sequenced reads following bioinformatic analysis are indicative of successfully cross-linked sequences. These mutations are T to C transitions when using 4SU and G to A transitions when using 6SG. The defining feature of PAR-CliP is the possibility to derive the location of the crosslink by scoring for a characteristic mutation residing in the cDNA libraries prepared from the recovered RNAs. This allows for the easy separation of the signal derived from specifically bound RNAs from the noise of sequences derived from co-isolated fragments of abundant cellular RNAs.
So we've shown you today how to isolate RNA segments specifically recognized by RNA-binding proteins. When doing this procedure, it is important to work RNAs free. So that was it. Thanks for watching and good luck with the experiments.